Welcome to my channel, Correct Biology. In this video, I am going to teach you how the peas, they are going to have wrinkled seed. So here you must be knowing, in the case of pea, you can have round pea. These round pea, they are going to have this type of shape. Yes, you can have wrinkled pea. The wrinkled pea, they are going to have this type of shape. They are not perfectly round. So in this video, you are going to learn how pea, they are going to have round phenotype, how they are going to have wrinkled phenotype. Now, we are going to discuss how things are going to happen at the molecular level. So here you have to understand the final P phenotype. It is being decided by a gene, that is SBE1. Now, if you see, this is actually starch branching enzyme. 1. So this is starch branching enzyme 1. And here I just want to tell you, if you have both alleles normal, so within the seed you are going to have normal starch branching enzyme, capital S, what you are going to see? You are going to see round seed, and if you are going to have this one heterozygous condition, capital S, small s. Here in this case you are also going to see round seeds, and if you are going to have this one small s, small s, means that this enzyme is absent. In absence of this enzyme, what you are going to see? You are going to see wrinkled phenotype. So one minute please. So here what we are going to see? Wrinkled phenotype. Now, how the absence of starch branching enzyme 1 leads to wrinkled phenotype, that we are going to discuss. Now, try to see. In the case of plant, you must be knowing that sugar, it is being transported, in the form of sucrose. So majorly you know that by the way of photosynthesis, the plants, they are going to synthesize sugar in the form of glucose, and later this sugar, it is being transported in the form of sucrose. And hopefully you remember that in case of sucrose, what you are going to see? Glucose and the fructose. These two monosaccharides, they are being joined together by alpha 1,2,2 glycosidic linkage. So if in between glucose and fructose you are going to have this alpha 1,2 glycosidic linkage, then you are going to have the disaccharide sucrose. Sucrose is disaccharide, and here in this form the sugar, it is being transported from leaves to other parts. So here within the seed, what we are going to see? This sucrose will reach, the sugar will reach in the form of sucrose. So let's suppose you are going to have these sugar in the form of sucrose. So here I am representing this sucrose molecules. Now try to see here, the plants, they actually store sugar in the form of starch. So the plants, they are going to store sugar in the form of starch, and hopefully you remember that starch is going to have two units. One is amylose, another is amylopectin. Okay, now try to see, this amylose is going to have sugar molecules, okay, joined together, okay, but here you are not going to see any branching. Okay, here what we are going to see, the glucose molecules, okay, they have been linked, okay, through alpha 1 to 4 linkage, okay, alpha 1 to 4 linkage, okay, the sugar molecules they have been joined, okay, the glucose molecules they have been joined, but here you are not going to see any branch points. In case of amylopectin, okay, here I am just drawing that the glucose molecules they have been joined, okay, by alpha 1 to 4 linkage, and also you are going to see branch points, okay, you are going to see branching. And who is going to introduce these branching? The enzyme SBE1. Okay, so here you see, in the presence of this enzyme, what we are going to see, starch will be synthesized. Okay, if this enzyme is absent, okay, what we are going to see, we are going to see only the synthesis of amylose, not the amylopectin. Now try to see here, come at this point, within the seed, the sugar, it has been transported in the form of sucrose. Now here, if this enzyme is present, SBE1, so this sugar, okay, it will be converted, okay, in the form of starch molecules. Okay, so here you are going to see that this sugar will be converted into starch molecules. Now try to see here, initially you were having more number of sugars, but later if you see by numbering, you are going to have few numbers of starch molecules. That is going to give the benefit. Here initially if you see that there are large numbers of molecules of sugars, but here you are going to have less number of molecules. If you are going to count, okay, you are going to have less number of starch molecules in comparison to the number of sucrose molecules. Okay, here the whole sugar, it has been stockpiled in the form of starch. Okay, now here you must be knowing that when you are going to have anything, let's suppose this is anything, okay, here if you are going to have these number of molecules of solute, okay, so what will happen if there is some semi-permeable membrane, okay, let's suppose this is going to have semi-permeable membrane, what is going to happen, water will enter here. Okay, why? Because if by chance here you are going to have more number of water molecules, the water molecules, they will move inside from its higher concentration to lower concentration. Okay, if you are going to have more number of these molecules. Now let's suppose, okay, you are going to have the same condition, and if all these molecules they form one molecule, okay, what will happen here, you are not going to see the same thing. Okay, all these molecules they get converted into one molecule. Okay, and hopefully you know that this osmotic pressure, okay, it depends on the number of molecules. So here when you are going to have more number of molecules, the osmotic pressure will be more. Okay, more amount of water will enter and it will create more osmotic pressure, and here what you are going to see, less osmotic pressure will be created. Okay, so osmotic pressure, it depends on the number of molecules. Now try to think here, let's suppose you are going to have the seed, okay, and within the seed you are going to have these, okay, starch molecules, okay. Okay, so here this condition, okay, here what you are going to see, these are being synthesized when you are going to have the genotype, let's suppose capital S, capital S, capital S, small s, okay, means here you are going to have the presence, I am representing the presence of this enzyme SBE1, okay. Now let's take another situation, okay. Here you are going to have the seed, okay, and here what we are going to have, more number of these sucrose molecules, more, okay. Now try to see here, in this situation when you are going to have more number of sucrose molecules, what is going to happen, water will enter. So water will enter, and these seeds, they are going to have high osmotic pressure, very high osmotic pressure. Now with the time of development of seeds, what is going to happen, these seeds, they will lose water, okay. Now when they are going to lose water, what will happen, earlier they were round, but now they will become wrinkled, okay. Now here in this situation, when you are going to have this sugar stored in the form of starch, what is going to happen, less amount of water is going to enter, and this amount of water is going to maintain osmotic balance, that is going to maintain osmotic balance. And here you are not going to have high osmotic pressure. With the development, what we are going to see, you are not going to see the loss of water, okay. So here the seeds, they are going to retain the water, okay. So there is no pressure from inside that water must be lost, okay. So in this situation, what we are going to see, the seeds, they are going to retain the shape, round shape. So hopefully you have learned in this video how Mendel pea, they actually became wrinkled, okay. So this is the molecular explanation how the Mendel peas, they became wrinkled. Or here you can see in the nature that you are going to see two type of seeds, one is round, another is wrinkled, and this wrinkled phenotype is due to absence of the enzyme SBE1, and I have explained you how this enzyme is going to code for a protein and how that protein is going to work like an enzyme, okay. So hopefully everything is clear. Thank you for watching this. Please do like, share the video and subscribe to my channel Correct Biology.